We are now in the basic view of the software, which is the view mode, and we are seeing an expression dataset obtained from the Cancer Genome Atlas. This dataset is composed of 150 colon cancer samples, which are grouped in 4 vertical columns according to the stage 1, stage 2, stage 3 and stage 4. And we are seeing actually expression levels through the heat map of 600 Kinome genes that have been selected based on an existing database and clustered by their expression. So we can see different expression clusters here. We can actually see the entire picture, and we can drill down to see each individual kinase in each individual samples and the expression level. The classical 2D heat map captures expression of genes across samples, but it does not capture additional molecular profiles carried out on the same samples. For example, the TCGA consortium generated copy number alterations also for the same genes in the same samples. And actually, to see this data together with expression data, we need to add the third dimension. So we can start exploiting the 3D features of GenomeCruzer, and we switch to the 3D mode, and we visualize copy number alteration as height of each single feature by increasing a scale factor. And so we start seeing individual genes in individual samples coming out at different heights proportionally to the copy number gain or loss. To better distinguish gains from losses of gene copy number, we can remove the absolute value feature so that gene gains stay above the floor and gene losses go below the floor because they are negative values. So in this way, we can also immediately see that there is a very good correspondence between gene amplifications and overexpressions and genes deletions and underexpression. To quickly switch between above and below the floor, we can actually use the flip function and simply see all deleted genes above the floor in this view. We can actually see individual genes, for example this one, and see also the values of height and color, that is of copy number and of gene expression. And we can go to see recurrent lesions along the horizontal rows. Another feature of GenomeCruzer is the front wall. The front wall contains a series of buttons, and each button contains essentially a list of genes belonging to a given group of interest. In this case, the buttons on the wall contains member of canonical pathways from the molecular signature database of the Broad Institute. We can see, for example, this button contains PIP3 signaling in lymphocytes and these other T-cell signal transduction and so on. We can actually see the individual genes on the floor, and to better see them, we actually have to remove for a moment the 3D feature. And then we can select a gene set like this one, and we can see these light blue lines crossing the floor. And we can also see that their distribution is non-random. To better see the individual genes, we can reduce the level of detail on the floor so that we can actually see individual expression clusters, and now the genes are more clearly visible. The distribution is non-random, and we can see the different gene groups are distributed differentially across the various expression clusters. And here we can see some examples, like in this case, cell cycle genes are actually concentrated on some clusters. Now, we typically would like to answer 2 questions. How can we systematically assess enrichment of gene sets in individual clusters? And how can we assess actually differential gene expression across sample groups? To carry out this type of analysis, we have to switch to a different mode of operation of GenomeCruzer, which is the analysis mode. We now enter a different environment always 3D. And now we still see the front wall, but the floor now is differently organized. Here we have individual genes belonging to the individual transcriptional clusters in a hierarchical mode. So we can see transcript kinase cluster 1 and subcluster 1.2, 1.2.2, 1.2.1, and so on. So that now we can zoom in from individual genes to a global view of their clustering. And on the side wall, we have groups of samples. That means groups of patients. Here we can see the stage 1 patients, stage 2 patients, stage 3 patients, and stage 4. In the analysis mode, we can carry out differential expression analysis. Suppose we want to see what genes are differentially expressed between stage 4 and stage 1. We use the differential selection feature and select group A as stage 4 and group B as stage 1. Now on the floor, we can see the color representing gene expression, and we can immediately see a cluster of genes which have a lower expression in stage 4. And the height of each feature of each gene represents the p-value, that is the significance of this differential expression. So we can see, for example, that JAK3 is strongly suppressed with a p-value below 0.0001. This cluster of genes with lower expression in stage 4 is very interesting, and we would like to know if this group of genes is enriched in any of the buttons of the front wall. So we exit differential selection and we select these gene clusters, and now we see that on the front wall each button becomes smaller or bigger, and the size is proportional to the level of enrichment. The color represent the differential expression of members of these groups across the 2 chosen samples for differential analysis, and the transparency indicates significance. So this group of genes, which is TCR signaling, T-cell receptor signaling, is strongly enriched. 7 times the delta is 7.1, which means they are 7 times more abundant than expected, and the p-value is 10 to the minus 6. We can also see other gene groups enriched together, and again we see T-cell receptor signaling. We can see that also other gene groups, like this one, which indicates JAK-STAT signaling pathway, and this one, which indicates chemokine signaling pathways, all these gene groups point to a lymphocyte kinase signature. And this kinase signature is clearly more expressed in stage 1 good prognosis sample than stage 4 poor prognosis sample. As you have seen, in 2 simple steps, we have carried out differential expression analysis and enrichment, ending up with a biological message coming out of this data. Another interesting way to use GenomeCruzer is to explore profiles of single genes or single samples. As an example, we can see here the genes with the strongest downregulation. We can actually select this sample, this patient, and now we can see expression of each members of the Kinome dataset across all clusters. And we can exploit the presence of a copy number alteration, and we choose the height dataset, and we can see amplification and overexpression of the same clusters of genes. We can then be interested in checking for recurrent amplification in stage 4, select stage 4, and we can immediately see one gene popping out here, and this gene is BRAF. And now we can look at the single gene, BRAF, and actually see that it is much more frequently amplified in stage 4 than in other stages. Finally, we can check whether BRAF copy number gains correspond to overexpression and switch back to color dataset and actually see overexpression of BRAF in stage 4, at lower rate in stage 3 and stage 2, and at much lower rate in stage 1.